honorary Pearsonite, so we're starting. But yeah, James and I have been ride or die since day one. It's actually 5 years ago at Bulldog Days that we met. We were at a presentation by Marvin Chun, and this guy, turns to me and says, "Hey, what's your name? Where are you from?" And we started talking about San research at UCSD, and kind of the connection just go from there. He's been really my best friend from Yale ever since. It's actually pretty funny. I ended up taking a gap year and living in San Diego. And the apartment that we lived at was actually the same one that James had lived at when he was a kid. And so it's kind of how the connections run so deep. Yeah, here I am to introduce James for his Vellum Forum. James and I have watched a fair amount of TV and movies over the years. And so I thought I'd talk about how we reflect some of our favorite on-screen dynamic duos. So first of all, we have, of course, Walter White and Jesse of Breaking Bad. James and I, like Walter and Jesse, really like to do science together. We've taken a science class together every single semester while at Yale, all 8 semesters. Our first science class together, one of our favorites, was of course freshman Orgo. Before every exam, we find a room in WLH or LC or BAS, and for hours just give each other problems and fill the boards, the chalkboards with problems. And really, we would never erase it because we always like to think about the person who'd come in the morning, look around and say what went on in here last night. Really, we made organic chemistry fun for each other. We also did organic chemistry lab together, and let me tell you guys, in the language of James Hahn, he is a weapon in the lab. Whereas Joseph Walensky would say he's got GT, a good technique. It was in organic chemistry lab that he got the most prestigious honor that is offered at Yale. I got the highest yields in organic chemistry pen. We found times to experiment outside of lab classes as well. Shout out to the Trumbull Dining Hall. One of the other favorite classes that we took together was topics in cancer biology, taught by Dean Chang. That's actually where we started calling each other doctor. We also that's also where we picked up the phrase astute observation. Speaking of language, James has a very unique lexicon and is definitely influenced my greatly. I thought I'd take a second here to cover some of the most important phrases that you need to know to understand James. Of course, there's big brained, adjective, which refers to someone who's making smart decisions, coming up with good ideas, or succeeding academically. There's a top 10 anime betrayal. This is a noun, which is an action that James does not like. I do have to say at this point, we're probably well past 10, maybe in the triple digits. And then finally popping off, which is just kind of a verb for succeeding at whatever you're working at. Another one of our favorite duos, Gon and Killua from Hunter x Hunter. Who love going on adventures together. And James is a very adventurous person and has brought me on many adventures over the years. We did that one day camping trip to the Catskills, barbecue festival where we competed, not very well, in a bacon eating competition. We did a marathon of sorts in Central Park. And most recently, we crashed a Brantford Formal after going to an Eddie's party. So James showed up to Barcelona in a graduation gown and a towel. With this wild and fun side, you might think that James, like Candace, is of the work hard, play hard mentality. But I would say that's not quite right. I would say James is more of a working efficiently and when needed, and then play hard mentality. For example, James would show up to our weekly 15-minute cell biology quizzes 10 minutes late and finish the quizzes within 5 minutes and score astoundingly well. If I had a nickel for each time he started studying for an exam or working on a paper by the time I had finished and still knocked it out of the park, I probably wouldn't need to sell my soul in January. On that note, I would like to take a moment to apologize to all the faculty members in MDMB and SNDS because I lured him into consulting. And this presentation may well be the last time he uses anything he learned in your courses.
Yeah.
All jokes aside, Dr. Han is one of the kindest, funniest, and biggest brained people that I have ever met, and I'm beyond grateful to call him my best friend. I can't wait to see how he pops off in the future and what new things we cook up together.
I think in his little speech, Saul forgot to mention that he's actually going to be a doctor. And I'm just going to be. Yeah. So, please have a clicker.
Oh, yes, sir.
So for my presentation, it's a little more boring than Candace's, but I'll be, in the words of my title, investigating the read-through transcription phenomenon with high-dimensional unsupervised learning. Basically, combining my sort of two majors in a way that allows me to graduate.
Yeah.
So for a lot of context, I thought like there's a lot of things to review before we can get to what I tried to do in my thesis. But the basic idea of central dogma of molecular biology is that we can go from DNA to RNA to the ribosome and to proteins, and this constitutes what the cell is actually doing. So anything that your body does, anything that your cell is doing functionally, originates from the DNA and has to go through this RNA phase to get to the ribosome and to become a protein. So the RNA, you can imagine as an intermediate where no matter what, it pass through the stage of what we call transcription to become proteins that enact functions within the cell. So transcription, as you can imagine, it basically controls everything that your body makes. And because of that, it's very tightly regulated. There's a lot of different factors that relate to when the body or when a cell goes through transcription, whether, and there's three stages here: initiation, so of those three stages, in this presentation, I'll be kind of focusing on termination. All three of these stages are very important for describing how an RNA molecule basically gets created within the cell. Termination is the idea that once a polymerase, shown here, attaches onto a DNA molecule, it starts creating RNA, and that RNA has to be cleaved and it has to end. Termination or RNA synthesis has to end and terminate properly to create a functional RNA transcript. So in eukaryotic organisms, the RNA transcripts has to also be processed before they actually leave the nucleus. And this whole sort of like cascade of events is very tightly regulated. And the entire focus of my thesis was when that regulation does not work well. So when termination fails, there's this phenomenon called read-through transcription, which is basically as the RNA is being created, instead of finishing and terminating and the RNA becomes a mature RNA transcript, the termination won't occur properly and the RNA will keep processing along the DNA. So these malfunctions create what we call downstream of gene transcripts. And these are basically any RNA transcript that has elements of the gene that is not supposed to be here. So for example, here, you can see that the gene is supposed to end right here. And instead of just ending right here, the polymerase keeps going and it creates an RNA transcript that extends beyond that end of gene point. So we actually don't know what these downstream of gene or DOG transcripts are doing within the cell. So one avenue of research that the lab that I'm in is curious about is what they're doing and how they're made. But I approached for my thesis this question from a little bit of a different perspective. So just a brief overview on what RNA sequencing is. So in general, DNA sequencing has become a lot cheaper throughout the years. So after 2001, or the early 2000s, when the human genome was first sequenced, the cost per kilobase or megabase of DNA has been steadily decreasing. So that just means that to generate the same amount of DNA sequences from an actual cell, it's much, much cheaper. It's much easier to generate that. And from that, we've also developed other technologies to be able to explore the cell from any nucleic acid perspective. So RNA, you can make DNA back from RNA using a really cool enzyme. And through that, there's a technique called RNA sequencing, which basically takes the RNA from any cell. And after you chop it up, so this is like the sort of general process of RNA sequencing. So once you isolate the RNA from a cell, you cut it up and you turn it into these small fragments that you can then sequence. And once you have these short reads of the RNA sequence, then you can align them back to the genome and you can try to explore what the cell is kind of undergoing as a function of the different RNA transcripts that are floating around within the cell. So one example is, let's say if you treat the cell with some stimulus, for example, like a drug, and then you see that an RNA for a certain gene goes up, you can assume that that gene might be interacting or might have some role in processing the drug that you've injected. So basically, the expression of RNA that you can get from RNA-seq will allow us to understand what is happening within the cell. So generally, like RNA sequencing data is very complicated, and there's a lot of like crazy math that goes on that people have come up with to understand how we how to like understand this crazy amount of data. But the basic idea is once you've cut up the RNA and you've turned it into DNA, then you can sequence every single individual piece. So each one of these pieces is around 150 nucleotides. And once you've read those, then you can align it back to the genome. So we have a reference of genome for a lot of different species that we can compare each 150 nucleotide sequence to and assume, oh, like this is pretty close to, for example, this red part, so we can map it to here. And once we have these maps, then we can build an idea of how many times this gene has been expressed as an RNA, or this gene has been expressed as an RNA. So this is the basic idea of RNA-seq. But my question here was, can we use this data to analyze this idea of dog transcripts or downstream of gene read-through transcripts? So quick segue, machine learning. Machine learning is the idea that given some data that we have, when we teach a machine, a computer, to understand some underlying association or some underlying principle within the data. So there's three main types: supervised, unsupervised, and reinforcement learning. Reinforcement learning is used more in robotics and stuff. So for our purposes, we're concerned with supervised and unsupervised learning. But within the supervised and unsupervised world, it's basically an idea of whether we have an actual truth that we can compare our data to. So for supervised learning, we want an actual truth that we can train our model on. So if we know that something is supposed to be class A and the model incorrectly classifies it class B, then we can give it a penalty. Whereas unsupervised data means that we don't have an actual truth. So for unsupervised learning models, we don't have whether something is true or false, something is A or B, and we still have to make some sense of that data. So for our purposes, because we don't have an actual understanding of what exactly a downstream gene transcript would be versus what is not, I decided to proceed with unsupervised learning. Okay, thank you. So unsupervised learning, from a general sense, you can clump these into three different groups, like that, and that generalizes to 3 dimensions as well. And the idea is you can generalize this to any amount of dimensions that you want. So what I wanted to do was to create new dimensions to analyze each gene in this sort of like n-dimensional data space, and to see whether we could use that data space or that vector space to classify whether something was a downstream of gene transcript or not. So preliminary results, downloading the data for it was like 30 gigabytes, so that took a while. Learning how to do all the RNA-seq tools also took a while, but it's like preliminary results. This is like, I hope there's like a full graph. Like I have to meet with my thesis advisor tomorrow. I'm hoping these are like 2 different groups, and this is like one group and that's the other group, but we'll see. But next steps basically to try this on different samples and to see if we can make the machine learn the difference between the 2 better, and to also figure out how to see what's the right answer, and of course also to write a research report, which has not been started. But basically knowledge. My interest in dogs kind of came from when I realized that I had to do a research for credit class, and I didn't have a lab to do research in. So I found this lab. It was very exciting. So many thanks to the Sites lab, and also to my 2 advisors in my different departments, and to John Lafferty for his excellent instruction of intermediate machine learning. Yeah. So.
Question for James.
Can you talk a little bit more about that graph? Like what it is, how you go from like n dimensions to a 2-dimensional graph, like all that.
Yeah, so this graph is, so there's a lot of different ways you can go from n dimensions to 2 dimensions. So some ways they preserve like distance within the n-dimensional space to 2 dimensions. This one is like, it's a style of dimension reduction called TSNE, T stochastic neighborhood embedding. So it's kind of like actually really complicated. Like I personally don't fully understand all the stats behind it either. But the basic idea is it finds like manifolds within like an n-dimensional space, and it like presses that down into 2 dimensions and displays that. The misleading thing about these graphs is any distance on this like 2-dimensional graph doesn't necessarily correlate with the dimension in the or the distance in the true like, n-dimensional graph. So if something's really far, it could appear closer. Yeah, it's a package on Python. Yeah. Yeah.
Are there like meaningful differences like between like samples of like different like obtained from different humans? So like if so you have like some learning or like your model has been trained in like one sample from like certain demographics, and can you like why would you use like unsupervised learning? Because there's some like results that you can obtain and like then you can kind of transfer it into different demographics.
I think the main choice for unsupervised learning, one, was that there's not too much like, it takes a lot of biological infrastructure to actually generate true positive and true negative values. So you have to individually quantify each one with like RT-qPCR. Like that takes like kind of a long time. So rather than that, I wanted to explore a way where we didn't have to do that. And then another like consideration was that the individual variance between samples will be a lot higher than the variance between the two like classifying groups that I had in here. So that a classifier that was trained across all samples would just identify different clumps within the samples rather than finding differences between like downstream of gene versus. One more question, Daniel.
Yeah, so can you explain kind of just like how we can use the results from your analyses to like how can they can be transferred to real life medical solutions or stuff, and like also the important. I know you mentioned that you were hoping to see like 2 distinct groups in the data you have. Like, what is the significance of like having 2 separate?
Yeah, so the main question with these like dogs is that we don't really know what they're doing. So it's hard to really say what medical impacts that they could have, other than that, like potentially they could be the cause of some diseases. There's like some research going on that suggests that they may be part of some immune responses. They might be part of like nuclear scaffold support after like viral infection. But other than those, it's hard to really say because these are widespread phenomena that are observed whenever a cell is stressed. So there's a few different hypotheses floating around of why they're actually generated. In terms of like the idea of like grouping the 2 different like clusters, I guess. Once we have a way of identifying from a big data set which transcripts are dogs and which are not, we can extend that to like lots of different data sets. We can like adopt like a high throughput approach, and you like combining that data in like a meta-analysis, we could then see if there's any sort of like correlation between these dogs and certain diseases, or like correlation between these dogs in a certain gene and like certain diseases. So that would be like the future of it, but it's like kind of far off.
All right, and finally, we've been waiting to do this presentation. It's going to be firmly known forum in late March.
Yeah.
Diego, as introduced by Lata. All right. All right, please.